And we are now recording, and it's all yours. Thank you, Eric, for agreeing to give the seminar.
Okay, great. So good morning here from California, and good whatever part of the day it is to everyone around the globe, and for those who are watching later online. So first, I want to thank you, Kate, for providing the opportunity for me to share some of our lab's work. So I'm Eric Wei. I'm a PhD candidate in the electrical engineering department at Stanford University, working in 𝗟𝗏𝗃 lab in bioengineering. And my primary research interest is building synthetic cells. So I've been part of Build a Cell kind of from the very start. I was even part of the group that made the first Build a Cell website, where we added the video that you see here. And Kate and whoever else have done an amazing job upgrading the website. But I'm so happy to see this video as part of the launch page. So anyone who's gone to buildacell.org and have seen this video, you can stay tuned and you'll find out what we're showing here. So I'll start by giving a kind of a snapshot of where we are in terms of building synthetic cells. So those in the field are very familiar with kind of the two different approaches, one being the natural, also known as the top-down approach, particularly the strategy of minimizing genomes in order to get a minimal genome of just these central genes. And also the synthetic bottom-up approach, where you are able to construct protocells that you hope to replicate genetic information and also proliferate spatially. And those have particularly, especially those working at abiogenesis, are interested in this approach. So if you compare the two, you have the top-down approach where you're able to operate on a fully functional live cell. So you're able to use all of the tools of biology, and also you're able to have a very easy readout on how you can tell if and use the cell that's working. But using this approach, for example, 𝗟𝗏𝗃, when they were able to have a minimized genome of 473 genes, but 149 of those genes, nearly a third, had unknown biological function, and 79 of those could not be assigned to a functional category. So you have a subset of genes that remain unknown, or essential genes of unknown function that we're working to understand. And for the bottom up, although we've done a lot, there's a lot of amazing efforts in a lot of the different parts that are necessary for a cell to replicate. Though all of those parts have not been integrated. So you have essential functions, some of which may actually be unknown, that you have to encode into the genes. So we have a lot of work to do in the cell building community to be able to figure out these mysteries. But in order to tackle this monumental task, I kind of want to frame it in terms of the message I want to impart to all of you in my talk today, and that how we organize ourselves will impact how we organize ourselves, and in part, how we organize ourselves will impact how we organize ourselves. So in other words, there's synergy and feedback on how both how we organize and build the community of cell builders and how we organize and build the cells themselves. So to kind of root this concept in my home field of electrical engineering and computers, I like to share a story of one of the first operating systems for computers, the Unix operating system. So in the mid-60s, Bell Labs partnered with MIT and GE to make a new time-sharing operating system. But after 4 years, the project was canceled. But the small team at Bell Labs wanted to continue working on this project. But all they could get their hands on was this PDP-7 mini computer, which at the time, definitely wasn't the cutting edge. But because of what they had to work with, they had to build a very simple, efficient, and composable system. And through the fruits of their labor, which they coined as salvation through suffering, which incidentally is what I coined the mantra for people who pursue PhD programs, they kind of guided a programming philosophy which they detailed in their manual. So I'll share a brief excerpt. Even though the Unix system introduces a number of innovative programs and techniques, no single program or idea makes it work well. Instead, what makes it effective is an approach to programming, a philosophy of using the computer. Although that philosophy can't be written down in a single sentence, at its heart is the idea that the power of a system comes from the relationships among programs than from the programs themselves. So they had a very conscious organization of the system that they made. But not only that, they also had conscious decisions on the community and the collaborations that they wanted to impart. So I'll let them speak for themselves on that front.
We don't hear the audio, if that's what you're
Oh,
playing.
you don't hear it? Oh, no.
Eric, when you set up your share screen, there's a box that you need to click in order to enable the audio to come across.
Okay.
You might have to drop out of share screen and restart screen share hitting that toggle.
This is awesome wallpaper, by the way.
Ah, I see. My fault. Okay, let's try that again.
Back in 1969, a couple of computer scientists here at Bell Labs started to develop some programs they needed for their own use. What Ken Thompson and Dennis Ritchie started developing then has evolved into the Unix operating system, which by now is widely used around the world.
We were trying to make computing as simple as possible. In the late 1960s, Dennis Ritchie and I realized that the then current operating systems were much too complex. We attempted to reverse this trend by building a small, simple operating system on a mini computer.
Well, what we wanted to preserve was not just a good programming environment in which to do programming, but a system around which a community could form, fellowship. We knew from experience that the essence of communal computing, as supplied by remote access time-sharing systems, is not just to type programs into a terminal instead of a key punch, but to encourage close communication.
So from their conscious design decisions then, you can see even to this day, if you go to unix.com, right front and center, you have a forum where you can discuss with other members of the Unix community. And this process works. As you see, nearly all of today's operating systems are derived from Unix. Basically every non-Windows operating system. So even if you're not familiar with Unix, I bet you use either iOS, Mac OS, Linux, Android, Chrome. So all of these were derivatives of Unix, which shows the power of being able to build a platform that enables collaboration, and how those collaborations bring fruit to a thriving ecosystem. So you definitely in this field have heard a lot of parallels from electrical engineering to computers to cells. But having worked in this field, having worked with cells firsthand, I don't want to make I don't want to declare that the way that we engineer computers, we can directly apply that to biology and cells. In essence, how we program and engineer hardware and software, we can't directly apply it to wetware. And there's many reasons for that. The main caveat is that cells have undergone billions of years of selective evolutionary pressure, and none of those pressures included being able to be human engineerable or understood. But because of this notion, it really drives my motivation for wanting to build synthetic cells for the purpose of being able to enable collaborative development by building cells that are simpler and understandable. So if we want to build a community of cell builders, we should be looking at, a minimal cell where we have a cell operating only on the core functions of molecules for life, which I introduced kind of as a top-down approach. But more importantly, we really want a minimized cell that can be modelable. So if we have a simpler cell where we have where we know all of the molecules, and be able to determine, model, map out the functions and how they interact, then we'll be able to have operational understanding on how to better engineer them. So many moons ago, I kind of brought this concept to Drew at a lab meeting. And during that lab meeting, he brought up the idea of something that may be the intersection of both of these ideas. So in the 1960s, they made a discovery of small anucleate cells that would arise during division of certain E. coli strains. So there's a lot of work putting into better understanding these miniature E. coli cells, are also known as mini cells. So, for example, this particular mini cell strain, which has a min-B deletion, causes random septations during cell division, which you can get miniature blebbings that occur at the pools. So we wanted to see for ourselves by getting this mini cell strain, and we grew them up, and we also saw that you could get these little blebbings when you grow and divide them. So there's also been a lot of different interesting tools and technologies that have been used with mini cells, and also a lot of interesting measurements. For example, what I'm showing in B is a cryo-electrotomography image of T4 phage infecting a mini cell. So these mini cells, which receive a portion of the cell milieu and machinery membrane, but lack a genome because of the size and the way they're formed, kind of brings the question, can we use a mini cell which has, kind of serves as a natural container, but bottom up synthetically build up the genome as sort of a hybrid approach that can utilize mini cells as a way to build synthetic cells. So if we wish to use mini cells for this approach, there are many questions that we need to have answered. So one, to use them, we have to be able to reliably transfer heterologous DNA into them. We're also needing a way to be able to purify mini cells from their progenitor cells. And does an individual mini cell have even the expression capacity necessary to express a genome in order to generate its own copy and replicate? So when we receive the mini cell strains, you see growth here under our microscopy setup. The first thing we did was we transferred a GFP plasmid into the progenitor cells of the mini cell producing strain, and what we saw was that, in these images, you can see that you can clearly see, especially in the GFP channel, you have all these bright hotspots that, call out that you're able to identify all these mini cells. So that really brings the question, are these mini cells expressing heterologous DNA, or is there another explanation for why we see accumulation of GFP fluorescence in mini cells? Could it just be maturation of expressed proteins that were from the progenitor cell? We have some colloidal physics experts in our lab that have other explanations for why they could congregate in these small spherical mini cells. Because the mini cells themselves don't dilute, they don't grow and dilute. They're kind of frozen in frame where you have your GFP signal just sitting there. So we need better ways to be able to characterize and measure, manipulate these mini cells on individual scales. So we want to be able to purify and produce them in high quantity, in high purity. So the way that we do that so we employ kind of established protocols that have been used to traditionally purify mini cells. So in order to have good differential between mini cells and progenitor cells, you can use centrifugation techniques to separate the two. So you first grow up a large culture of the mini cell producing strain, which produces mini cells. Then when you pellet, you'll be able to pellet most of the progenitor cells, leaving mini cells in the supernatant. So in order but in the supernatant, you also have a population of small growing progenitors that you'll need to remove. In order to do that, you grow out the supernatant to prepare for utilization of a antibiotic, penicillin-based antibiotic that causes cell lysis when the progenitor cells grow, but the mini cells themselves will be intact. And you can also, so our purification process yields, 6 times 10 to the 8th mini cells per 1 liter starting culture, and a final purity preparation of 500 mini cells per remaining viable progenitor cell. There are also other ways that you can improve the purity of your preparation, for example, through FACS or through storage in environments where the progenitor cells won't thrive. But so then we're able to now have a population of mini cells that we're able to probe further. So in order to answer the question whether mini cells are capable of heterologous gene expression, first we grew up the mini cells with the GFP plasmid in the progenitor cells. And then at the start of the mini cell purification process, we added a translation inhibitor, specifically chloramphenicol. Therefore, at the start, when you purify the mini cells, when you add the translation inhibitor, any accumulation of GFP signal would not be due to new synthesis of GFP off the plasmid inside the mini cells. But if you do see, but what we found was that when we compared the mini cell preparations that included the translation inhibitor and did not include the translation inhibitor, we had twice as much accumulation of GFP fluorescence in the preparations that did not have the GFP fluorescence, or that did not have the chloramphenicol. So we conclude that there was heterologous expression in the batch of mini cells. But again, the question that I posed in the beginning, can individual mini cells are they able to have the expression capacity necessary to instantiate genomes and replicate? So in order to better answer that, we use microscopy in order to track expression of individual mini cells. First, using purified mini cells from the process that was described, that I described. And so we used time-lapse microscopy in order to follow the progression of expression of GFP fluorescence over a couple hours, and we saw from individual minicells that there was a dispersion of expression of GFP. So we were able to use the standard curve in order to translate the GFP produced to peptide bond synthesis in order to measure expression capacity in minicells. Similarly, we used the same similar method to trace progenitor cells that produce minicells and track those minicells that were freshly formed by progenitor cells. Of course, using this method, we also had to take into account possible GFP maturation that occurred. But using this, we were able to get a similar measurement of peptide bond synthesis, expression capacity from minicells. So at the higher end, we could have minicells that exceeded 1 billion peptide bonds produced within this 2-hour time frame. And what's interesting is that this small proportion of minicells, about 13%, would be able to, if they were able to do that, then they could be able to fully express an E. coli genome in its full proteome. And taking this into account, there was a lot of work done previously to study how E. coli conjugation works and how that functions in minicells. They were able to show that you can get DNA, genomic and plasmid DNA into minicells using conjugation. But a lab in Japan reported in 2018 that if you're able to have 2 separate E. coli strains that conjugate half of the E. coli genome each, and have that recombine in the minicell, they were able to get transformants that were able to be able to grow from the amalgamation of these half genomes into a full genome. So the recovery efficiency was really low, as you can tell. You have to have a lot of things work in order for this to even be possible, especially even having the right minicell and the right expression capacity. So they will only get 4 transformants or 4 colonies out of 10 series experiments, totaling 10 minicells. So this happens at a very low frequency, but the possibility, but our results, and including this result, support the idea that minicells have the capacity for instantiating, housing, and expressing whole genomes, and even growing from whole genomes. So this idea of, if you have this container where, traditionally we've been utilizing cells and methods that alter, minimize, refactor its lineage-based genome. If we're able to kind of shed its original lineage and instantiate lineage, a ab initio designed genome, which allows us to have access to many of the traditional engineering tools, have asynchronous distributed collaborative development of genome that can be lineage agnostic, and therefore instantiate that into kind of this lineage-based container, but have a lineage agnostic cell that's able to express off a synthetic genome. So that would be, so minicells would be able to be way, a method to allow for collaboration, collaborative development and community building within the building and use of synthetic cells. So this idea of Minicells as a container, you know, have an anucleated vessel. Minicells themselves have a lot of pros and cons, but you can also extract this idea to for example, maxi cells, where you're able to eliminate the genome of for example, 𝗏𝗏 or other cells that you may want to use. I think zombie cells is the term JCVI uses, or you know, 𝗏𝗏𝗃 labs sinels or 𝗏𝗏𝗃𝗃𝗃𝗃 or whatever you want to use. No matter what container you have, how are we able to design and prototype the genome we use, and also whatever container we decide to use that has design strengths and weaknesses for how we go about instantiating and prototyping the genome that we want to design. So if we go through this idea of lineage-agnostic cell design and building of the genomes, we're afforded to use, we couldn't use genes and proteins from you know, all forms of life. Of course, you know, we may not want to. You know, the cost is, you know, all the enzymes or all the proteins in bacteria have been co-evolved to work well together in a defined environment. So if you mix and match, you know, there could be potential detriments, some side reactions that are anticipated, but using an lineage-agnostic approach, you're able to have the option whether you want to use it or not, to be able to call from any sort of living resource from any living lineage, or even like xenobiology, if that's what you want to employ. So an example of a system that you may want to encode is transcription translation, since it's a very well characterized, well established, well studied essential cell processing. And using this knowledge that we have, in Japan in 2001, a group was able to take all of the enzymes that they needed for all of the processes that were required for transcription translation, and build a fully defined reconstituted cell-free system known as the PURE system. So the PURE system, again, it's fully defined, and what I've listed here, it uses, you know, mostly from, it's color coded with the, for example, the orange is using E. coli enzymes and parts, but it also takes from, for example, myokinase from yeast, the T7 RNA polymerase, creatine kinase from rabbit. So they were able to utilize enzymes and parts from many different lineages in order to make a working system that they designed. So in terms of what PURE can exist as, in the US, where you can buy it off the shelf from NEB, known as PURE Express. I believe in Europe and Asia, you can, PURE Frex is the more prevalent commercial brand. But you can also make homebrew PURE by either individually purifying the proteins, or also there's been a lot of recent work, for example, like Qiming Tan's lab, that's been able you're able to have cheaper, easier ways of purifying PURE for labs. So PURE can also be an accessible tool for many labs as well. And also, because of its fully defined nature, you can add or remove certain proteins, and many labs and researchers have done so for whatever you want to use or study in the PURE system. So as a fully defined system, there's a lot of strengths. For example, fully defined systems enable development of fully defined models. So in the lab that produced Pure, they wanted to develop a model, for example, where they modeled the synthesis of this tripeptide I'm showing, and they had and they tracked and they modeled 968 different reactions of 241 different components. So since all of the individual components of Pure are defined, you can have a model that can track and have and model each interaction that exists in the system. If you have a fully defined system, you're also enable the construction of fully encoded genomes. So, for example, the paper which was, I believe, one of the first papers that were reviewed in the Build a Cell seminar series, they were able to encode different parts of the Pure system into 11 different plasmids, totaling 116.13 kilobases. And they were able to co-express some of these plasmids and show that for some of the components of the Pure system are able to have expression greater than the amount that already exists in the Pro system. So sort of like a gain of the components that can be produced in Pure. There's also been a lot of work in this field in terms of encapsulation and gene expression of Pure in liposomes. That's an important part of being able to build cells, to be able to in space grow and divide them. So, for example, Christoph Danilon's lab in Delft, they're able to they've done a lot of work with Pure, including synthesis of membrane proteins, and also synthesis of lipids within the Pure system in liposomes. So we've done all this work in terms of working with Pure and cell-free systems, but how are we going to integrate, the genome design, the Pure system, the encapsulation and grow and divide? So there's a lot of work that we still need to employ, and also a lot of mysteries that or targets that we need to, objectives that we need to fulfill in order to achieve this. So one of these kind of dark, murky clouds that exist is the idea of, are we able to genetically encode a self-reproducing system? In other words, can we design a Pure self-free system to make itself? So the kind of to try to, in terms of a equation, if we have the Pure system and a genome that encodes the Pure system, in order to successfully build a synthetic cell, we're going to need to have the system make copies of itself and the genome, and so it has to fulfill kind of this quantitative condition of making making a copy sufficient to what it was, and also this qualitative condition that if you kind of expand this, if you duplicate Pure, then we know that Pure itself, all of its enzymes and parts can be functionally made in living cells that we then take and use as our self-free system. But it's unclear whether the Pure system itself can functionally make the Pure system. So in other words, a milestone achievement would be if we can have a Pure self-free system made entirely by the self-free system itself, all the enzymatic components. So this idea of kind of whether Pure can self-produce Pure, it's been explored a lot in the recent years. So for example, in Junli and George Church's lab, they were able to express the 30S subunit R proteins in pure, and coupled with the 16S natural 16S RNA and natural 50S subunit of the ribosome, they were able to recover about 13% relative expression to the natural full 70S ribosome. Interestingly, when they added chaperones to the system, they were able to increase that number. Similarly, just these past few months, there have been papers that been able to show that you can have that they investigated pure expressed tRNAs. tRNAs in particular, natural ones require a lot of base modifications, in which a lot of these processes and enzymes are not included in the pure system. But yet, without these base modifications, this group was able to show that you could have 40% recovery of expression when utilizing pure synthesized tRNAs versus natural tRNAs. And about a month ago, there's also a paper that using an approach where you utilize microfluidics, where you kind of flush in the full pure system, but then flush out pure system, where you have a full pure system that makes a single component of pure, but then flush out and introduce a component without the system you're testing. If the system was able to produce the initial component, for example, T7 RNA polymerase, they wanted to see if you could get regeneration of the pure system of expression using the missing component that was then encoded by the component that was made by the system itself. Okay, so all of these use a similar strategy. So I'll be expanding on how to go about if you want to be able to individually validate an enzyme in pure for self-production. So for example, if you have the alanine TNR synthetase, if you have a mixture of pure that contains alanine synthetase and does not contain the alanine synthetase, and you want to test the alanine synthetase produced by pure works, then first, if you compare the expression of a GFP reporter with and without the alanine synthetase, we found that you get 0.02 relative expressions, so 2% expression levels of GFP pure without alanine synthetase compared to pure that has alanine synthetase. So then we want to be able to get alanine synthetase that's produced by pure, but of course pure itself also has alanine synthetase within it. So you have to have some sort of method to be able to express and then purify the alanine synthetase that was produced by pure. So particularly we used a method where we expressed alanine synthetase with a strep tag, and we're able to purify alanine synthetase that we know that was produced by the pure system. So then when you add the alanine synthetase produced by pure in the pure deficient of the alanine synthetase, or the dropout pure, we found that when you added, that the pure system was then able to recover expression. So it was able to have about the same level of GFP expression as the full pure system, but now using the alanine synthetase produced by pure instead of the alanine synthetase that we got from E. coli that we put in the pure system. So it shows that pure can self-produce alanine synthetase as a subcomponent of the system. So then if you want to abstract this process out to all of the single enzymes in pure, the process you would have to go about would be to first make and test each type of dropout pure, be able to express your pure, express the whatever you want to test in pure and purify, and then add the purified system back to pure and then measure the recovery. So in order to get these dropout pure kits, first we wanted to, basically we had to individually purify. So when you buy NAB Express, it comes with all of the components mixed together. So if you want to be able to purify, or if you want to be able to get a pure system without a single component, then you have to be able to individually purify them. So for example, from these are 20 different synthetases that I individually purified, and luckily we were able to show that when we put it together compared to the purified synthetases that we received from NAB, we were able to get almost full expression recovery using the purified syntheses we had. But then, to expedite the system, also there's some particular proteins that are particularly hard, or at least in my hands, to purify in from pure. We were able to get dropout pure kits of individual proteins specific from pure. So basically, all the synthetases we were able to make dropouts, and all of the non-synthetases we were able to get a NAB provided pure kit without that individual. So in total, we wanted to test the dropouts for all of the pure kits. And interestingly, even though all of these enzymes are critically essential for gene expression, we found when we tested these single enzyme dropouts, we saw variable amounts of expression. And in order to use this assay, we decided that we would use, if we were able to be able to get recovery from a dropout that was sufficient that had 75% expression compared to the full pure system, then we'd be comfortable saying that we were able to have full self-protection. So in order to illustrate our measured self-production of all these enzymes, we came up with a kind of box where the top left, we record the depletion. So if you remember the 2% from the depleted pure for alanine synthetase, and the recovery for when we add the pure complemented component, for example, the 108% from alanine synthetase. So basically, the easy way of reading, the readout would be blue means that the depletion assay is working and recovery assay is working. So that's the most ideal. While the more red means it's not working, and more green means that it may be working. So to give other examples of again, for example, IF2 that we saw that when we dropped out from the pure kit provided by NAB, we don't get depletion. And for histidine synthetase, for example, we get partial depletion, and also when we try to provide the histidine synthetase produced by pure, we are able to get near but not full recovery. So we saw we had variable capabilities for self-production depending on the enzyme we tested. So then I show right now where we're at in terms of trying to flesh out this periodic table or measure self-production of individual pure enzymes. So our goal is so we've done about half of the enzymes, about our goal, or at least half of the remaining ones that we want to test. And coupled with what I showed earlier in terms of kind of what the community has already been able to show for self-production of pure, we're setting out to complete, to show where we're at in terms of what pure is able to self-produce. And this is kind of a call to action for the community, since there's a lot of work in terms of improving the capacity of pure to make pure, testing how done, again, quantitatively. There's how do we get pure to make pure, to increase the expression capacity of pure. So again, there's a lot of opportunity for collaborative development in terms of the making pure better approach. So again, what I want to part is in terms of the build cell community. Every time, every single build cell summit or every build cell meetup that we've had, I feel like we're getting better. We're doing more and more. But again, just kind of, I just want to instill to everyone who in this community, how are we better able to collaborate, to work together in terms of how we are able to enable our own ways of designing, building cells, and in part how that will form the community that we build, and how that feeds back into one another. So in terms of collaboration and thanks, I definitely want to thank a lot of people for making this work possible, especially particularly in Drew's help, particularly in our lab, Anton Jack Smith for a lot of the Minicell work, Connery Meyer, who was instrumental in getting Pure makes Pure, getting that up and running in our lab, and now he's doing great work in Qiming Tan's lab. Also, in terms of the build-cell community, a lot of these ideas have been fleshed out with people in the cell-free Pure makes Pure working groups, particularly Vincent Leroux, Lauren Williams, Luis. It's been a big help in formulations and iterations of these ideas. And then again, the steering committee, particularly Kate, they've been doing a tremendous job in terms of setting up, kind of building and increasing the momentum of the Build a Cell initiative, and also even in terms of what the world's going through, be able to still carry that momentum over. I'm greatly appreciative of that. And also in terms of NEV, they've been a great help, be able to make our customized orders. So I also want to thank them. So thank you all. And yeah, I'm happy to take any questions that there are. So thank you.
Thank you very much, Eric. That was really awesome. Periodic table is awesome. So if people have questions, you can unmute yourself or you can ask in chat. I have kind of a first question that might be, I guess, a little pushing the boundaries of it, but what do you think, how much could you minimize this system, do you think? So we can, meet with the current abilities of copying genomes in cell-free, which, it's been shown that you can copy 40, maybe 50 genes. Can you minimize translation system?
Oh, sorry. Yeah. So the way I see it, the Pure system itself is already pretty minimized, especially if you compare it to cell-free TXTL. Of course, the caveat, like you mentioned. So I just said the 56 genes. I think that's been shown in cell-free, right? But in Pure, yeah, that's still like really difficult ways to get that much gene expression. But I think we know from cell-free systems from living systems that it is possible to be able to reach expression capacities that are able to instantiate genomes. So I think even though the Pure system is pretty minimal and pretty crappy, but the way we should look at it is building up from that, using the knowledge that we have. And there's been a lot of work for be able to fully characterize cell-free systems in bacteria. So maybe if we fully know what's in bacterial cell-free, then are we able to then build up, maybe we have to add, and we showed the Junli's work with the addition of chaperones. Maybe there's has to be some sort of even different concentrations, different organization within Pure itself. So I think there's a lot of room to work towards. I think we should look at it basically making Pure better instead of stripping down Pure that's already pretty stripped down. So.
You have a question in chat.
Okay. Sorry.
I can read it out. It's
yeah.
You're talking a lot about the qualitative renewal of Pure, but just a question about the quantitative. So what's your current estimate in terms of, does Pure have enough peptide bond formation capacity, or how far away is it? And to what extent should the community be thinking about prioritizing Pure renewing Pure in terms of just energetics and cofactors, or do you think the better way to do it is as has been explored, sort of extrinsic exchange with fresh media, fresh buffer?
So yes, good question. So I didn't really touch in terms of quantitative, but that is a major problem in terms of what you get from NEB Express, the expression capacity of off-the-shelf Pure. For my submission, that's 30 times. You have to have 30 times as much expression capacity in order to have Pure have even the capacity to make itself, basically, to make all of its components at the concentrations of the Pure system. So that's already, far off. I think talking with Vincent Earl, in terms of cell-free, I think it's only about 6 or 7x in terms of TXTL systems. So there's, again, more we can learn from TXTL to better increase the expression capacity. But I do think that in terms of moving from a batch Pure environment to more encapsulated free exchange systems, that will be necessary in order to achieve a self-replicating Pure system. So any efforts that are able to increase the expression capacity of Pure, either by swapping out its metabolism, be able to better renew its resources, or be able to identify what's breaking down in Pure. I think there's a lot of papers that have investigated that, but I think definitely more work needs to be going to what's being produced in Pure that makes it break down so fast. And I think that will go a long way increasing the longevity and also expression capacity. So I think there's a lot of work in both qualitative and quantitative that have to be achieved.
And you have another chat question. Can you see chat, or?
Sorry,
I can read it to you. So great talk. I have a maybe somewhat naive question, but is your E. coli minicell entirely lineage agnostic? Are you sure there is no parental deposition of proteins, DNA, RNA into the minicell that will interfere with functionality of the genome you want to insert in it?
That's a great question, and also so I would say that in terms of minicells or any sort of I guess maybe minicells or any sort of container that you're derived from a natural cell, the minicells themselves are definitely lineage based. They're derived from a lineage. So if you can design lineage agnostic genomes in terms of genomes that aren't derived from a lineage. So it kind of combines the lineage based minicells that we know work we know that they're derived from a working living system to a system that we designed that we hope it works. But of course, if you use, and I'm definitely not prescribing that minicells are definitely the best way in order to fully realize the synthetic cell. But if you have minicells, you definitely have the possibilities of using a system that may not be well understood or may not be fully understood, or you could have if you have a more complicated system that's derived from E. coli or a full living system, that you could have some sort of unintended interactions with whatever fully synthetic genome that you have. But I think that the lineage-agnostic portion is particularly for genome design and specifics to ab initio and have asynchronous distributed development of genomes. I think that we can definitely be able to do with using minicells or other containers.